Okay. Okay, welcome everyone. It's a pleasure to be here and to welcome Karlina for her seminar. I'd like to start with an acknowledgement to the Iragunji, the Jabagai, the Gimwe, Wollabara, Yidinji peoples who have been caring for this region for many generations. This was and always will be their land. And we pay our respects to elders, past and present, and extend that respect to other Aboriginal peoples and Torres Strait Islanders who are present here today. So it's a real pleasure to welcome Karlina for this seminar. She's a conservationist, a conservation scientist whose research brings together technology, advanced data science, and community engagement to address complex environmental challenges. That work spans biodiversity, conservation, environmental governance, wildlife trade, human-nature interactions, and in particular, ecoacoustic and soundscape ecology. And that sounds really interesting. Looking forward to hearing about the ecoacoustics. Karlina completed her Master of Science in Ecology at Kanazawa University in Japan, and then her PhD at QUT, where she looked at economics of Indonesia's songbird trade. She has worked across academia, conservation organization, and government-related research, including as a researcher at QUT and Griffith University. And she's now working as a postdoctoral researcher here at James Cook University, and she's a key member of distinguished Professor Bill Lawrence's Managalas project team. She's published widely, and her recent work includes research on Indonesia's songbird trade and the developments of a typology for Australian terrestrial bird communities. And she's been recognized through a number of awards, including scholarships, QUT Outstanding Thesis Award in 2021, University of Queensland's CDCS Grants for Science Communication, and awards for her conference presentations. And please join me in welcoming Karlina Indraswari, who will now speak to us about making sense of large-scale acoustic data sets in the complex tropical landscape of Managalas. Welcome, Karlina.
All right, thank you very much. Thank you, TESS, for having me. So I'll talk a bit about the work that I'm doing right now with Bill and the team. It's a huge project. As you can see, there's so many people and institutions involved up there. And also a bit background of what I've been doing in the past as well. Some of these slides are kind of they're new and they're for sharing only. So if you see the sign of no photos or no sharing, please, I would appreciate it if no photos or sharing of these images on the slides are done. So I'm just going to move forward. We did an acknowledgement of country in the beginning, but I would like to extend that acknowledgement over here with the Jabugai, Yuranji, and Gimoy Yidinji peoples in Cairns. Even though I work with JCU over here in Smithfield, but I'm actually based in Brisbane, and those are the Turrbal and Yagara people, and the people of Managalas in PNG, in which we gather the data and do a lot of this project in Managalas as well. So a bit about me. Ages ago, I always had a question, how can we solve conservation problems, especially when they are complex? And then also in that journey, I, like everyone else, had a bit of a midlife crisis or quarter life crisis and thought, oh, I'll just have kids and live a happy life. That's not really the case, because then I always have a wondering, go back in conservation. And that's when I kind of shifted away from field work, going into more data analysis-based work. It was really eye-opening, especially since it did allow me to spend more time with my kids. And I started that first working wildlife trade with Traffic, which is an NGO, global NGO, but based in Southeast Asia, looking at a lot of trade issues there. And it was that extended me to my PhD. So hopefully those online can hear this. So if you heard that, you would imagine those are the call of some of Southeast Asia birds. You'd imagine they're beautiful birds in wild, but no, the reality is when I did my PhD that most of these birds came from those in cages. So that was one of the wicked problems that I tried to solve during my PhD. It was like, what is the best way to solve the problem of the legal trade? It was very complex because it involved a lot of people in Indonesia. I published a few from that. Some papers are also coming. And I was very, very fortunate to actually be part of a documentary by BBC producer Steve Greenwood, and called Endangered, where they actually documented part of this trade. And I was featured as one of the people there. And then after that, I did a postdoc where, because I love data, and it was really fun to look at patterns, we took a little bunch of data from Australia's bird data, which is citizen science, and found that apparently when you only look at the patterns of the bird themselves, Australia actually has 7 community regions and around 29 typologies. And we found that they're very distinct for birds. You'd have northern Australia birds, eastern birds, inland, southeast inland birds in Tassie. And then you'd have, southeast woodland birds, arid community birds, and then mallee and heath. So this has been published as well, and also some work is also ongoing and looking at degraded communities for this. So this was prior to JCU. I did a bit of those work. Happy to chat more about it if you're interested after. And a bit more, one more that I wanted to show is I'm also part of a team in Indonesia and East Kalimantan where we try to look after wetlands. So there's this piece of wetland over there that's been adjacent to oil palm and facing many threats of deforestation. Unfortunately, it's not protected under Indonesia law, and it is the only place in the world where the Tomistoma schlegeli, or the false gharial up there, and the critically endangered crocodile siamensis occurs together. And so I work with a team down there and they do a lot of spotlighting. I've been here in the past and I'm just kind of following up with what I've done before. And then after that, we also found this really unique ecosystem because we'll find new things like the flat-headed catbird never had records over there. And we found in camera traps that there were quite a few individuals and there was even a breeding population. So the one at the bottom left is that cutie little kitten that was captured in our camera trap. We've got orangutans, we've got stormy storks and proboscis monkeys, all endangered in the landscape, and also on the snake that on the bottom is actually, it's not the species there, that's just the genus. I'm not allowed to share the photo, but there's likely a new species that lurks around in the area that we work. And we are trying to get some funds to do work on that on the ground. So this is also, there's a research station and everything. Amazing people there on the ground. So if anyone's interested to go in there or do some research or bring some funding, let me know. Okay, so enough from my past and work. I want to take you to my current work, which is over in Managalas landscape. Very gorgeous landscape over in PNG. PNG is notoriously known for being very unknown and undiscovered, and we're about to enter territory that many people have not yet explored. But why Managalas? Why is this area very special? Is because Managalas is one of the special areas in the world and the biggest in PNG for a community-based conservation area. What does that mean? It means that this conservation area is governed bottom up. All the decision-making, all the protection, participation comes from the community to then protect this around 15,000 sorry, I had the number in my head, 3,600 square kilometers of rainforest, sorry about that, of very much a lot of untouched, unknown rainforest. This area does not come protected easily. Our partners in Landscape Alliance have been part of the constant years and years of engagement and continue to engage with the communities there to ensure about the protection and make sure that it remains to be conserved. So it also makes sense that the conservation and the monitoring of this area of the landscape also comes from the locals. So it's the locals that themselves have gone down, deployed camera traps, and deployed acoustic recorders to find out what's going on. So there's some images over there. And the landscape isn't really flat. It's very hilly, very topography-wise, very variant. So you can see there on the left side that that they're deploying in an area that's very steep. Access is also difficult, so that's Bill taking a picture of crossing the river when it was in dry season. So it's wet season, it's probably inundated. Then if, for those in the room, Danny Nane in the back there has been to the area. So if you want to know more about what it's like to work there, feel free to have a chat with him. So we're talking about acoustics. So let me take you to how it sounds like in Managalas, the data we have. That's a glimpse of what it sounds like and what I have to listen to, which is both gorgeous and also a bit daunting because this is what we soundscape researchers deal with on a regular basis. So those sounds, this image is going to be a bit laggy, but those sounds, if you display it, will look like this. Try to see if you can catch the sound with the image in the spectrogram. So that's a spectrogram, which is down there. On the x-axis is time, and then on the y-axis is the frequency. Yes, so that image, how do we even start to work with such data? Well, first we do something called annotation is very common. So whenever we see a type of call, we'll put a box around it. So if you go back to the, if we go back to the beginning of this, you can see how every call type, you kind of can distinguish, your ear and your eyes work together to find out where they are. And you're like, oh, that's one call type. It's a different call type. You go back and forth and try to put boxes around them to figure out that's what this one sounds like. And then we try to identify, is it a bird? Is it an insect? Is it a frog and stuff? This is something that I've done really quickly for the presentation. But even in this state, you can see there's a lot that I did not annotate. You can see in the bottom right over there that there's a kind of beep, beep, beep sound. I did not annotate that. And then you can see around the 4,000, 5,000, 6,000 hertz area, that's actually constant insect calls. I did not annotate that. And if you go above, there's other small calls. It's just so much going on in a single soundscape. So this is what we're always dealt with. And that's one few second spectrogram. Oh, sorry. This one here, let me see. That second one there, actually, it's even more complex. And this is calls from the dawn chorus. So you can imagine that every morning if you wake up, you know it's the most active where the animals start to sound and make calls. So that's the dawn chorus. And looking at that, I'm like, what the heck? Where do I even start to work with this if it's so complex? Now, that's a snippet of one area. In the Managalas project, this is a map of the Managalas Conservation Area and where we put these recorders out. Here we have 18 grids. And you can see the grids in the yellow and the red colors up there. And from the 18 grids, we have data from 4 grids so far. And we hope to have the remaining 18 either by the end of this year or early next year. For each of the grid, we have 7 by 7 sites. And each of the site means one recorder. And each of those are 500 meters apart. So then we have 4 grids, 187 sites so far. 1 minute every 5-minute samples, and looking at over 1.3 million minutes, or around 288 minutes per day. And this is kind of my reaction when I heard of that, both excited and very terrified. And then when I found out that that's only 4 grids, I kind of went double worried. And then we actually, for every single site, we also have camera trap data that we're working with. So yeah, that's kind of my feeling sometimes. So throughout the presentation, there's going to be some sound. I just want to actually put the sound on because it's always fun to listen to calls. So going into data analysis, these are some species that we found in our site. We've got the purple-bellied lory. And sound is so cool because you hear it, but when you see it on the spectrogram, you hear that frequency going up, and it just always fascinates me. And then our yellow-breasted boatbill. Is a completely different call. So how do we even start to work with 4 grids and that many sites per day? We're very fortunate that we have partners over at WildMon that has helped us with a machine learning recognition model, which they the one who find the calls and then try to find the patterns to match them. So going through those 1.3 million minutes, we're not done manually, but they made an AI recognizer to go through and try to detect what species are where. Excuse me. And then there's something else called acoustic indices, which I will get back to. But for now, we're going to look at how do they actually do this? Can someone replicate the method if they wanted to look at such data set? This might look like a complicated approach, but I'll try my best to explain it. So what happens is how it works with their data set is first they will take data and they will upload it. And then they use something called the foundation model, or we've got so many tools available online. And what it does is it detects sounds. And how do you detect sound is if you can see the spectrogram down there, you can see that calls have kind of darker colors. So those are usually higher in amplitude. They have different sound properties. So these are the ones that are automatically detected, and every single one has unique characteristics. So then you can develop like a model that detects these calls, and then they match it with the database that they have already. We're very fortunate that people in WildMon have their own database, but then we also are in a time where there's a lot of databases available online with a lot of information gathered from citizen science and other kind of surveys. So these are things like Xenocanto, if you know of it. They have a bunch of audio calls for animals, iNaturalist, and so on. So what happens is the guys in WildMon, they first go to another database, which is a GBIF database, which shows which animals are likely, which species likely occur where. So kind of doing a radius around Managalas, finding what species are likely to be found around that site. And then they'll have a list of species. From that list, we'll go on to the online databases and their own and find the calls. And then they'll have a template of what it looks like, and then run the AI recognizer through the audio and say, can you please find what species have these calls that look like this in our list and identify them. And this has worked quite well because they have around 32,000 detections so far that we have, and around 134 species. And how it looks like is on that spectrogram in the bottom there. Again, it's still boxes drawn and then identifying what likely that species is. This is also another excuse to show pictures and calls. So let's go through what we found. First is we have the Papuan eagle, we found most of the species that were found were least concerned. But we have some vulnerable birds, which is first the Papuan eagle. So that was quite rare. We had 26 detections out of 9 sites. Another vulnerable was the Paskit's parrot. As you can see, it's amazing call spanning so like really wide frequency bandwidth. I want to show this guy because there's been a lot of camera trap images of this species, the palm cockatoos going down to the forest floor, which is cool. Also, that one there, that's also from our camera trap, the Papuan eagle. So. And of course, being in Papua, our lovely bird of paradises. The Ragiana bird of paradise, the king bird of paradise. Magnificent. And our lovely streak bowerbird. So those were 7 from the 134 that were detected. Okay, so now we have those out of the 32,000 detections over the sites. How do we make sense of these patterns? So one of the simplest one that I looked at is, okay, this work is still ongoing, but the simplest one is, okay, let's just work on number of species and also diversity. From one site, which we call over there grid 38, this is a sample from those particular sites, and over there next to it is a heat map, and the darker color is, the more species diversity of that area. So we can see how even though they're 500 meters apart, we do have heavily diverse areas, the many dark places squares that we see there, but also right next to it, we have a kind of a lower diversity in the yellows. And these are 500 meters apart, which you would think that you can hear sound from that distance, but because the landscape is so dense, it's a tropical rainforest, and then topography is also very hilly, the sound may likely not travel as easy as we thought. So even 500 meters apart, there's already high diversity going on in those sites. Now, this is just a bit of me playing around with the data set. First, I also wanted to know then, if we're looking at measures of diversity, can we know how well they are partitioned? Meaning, in an area that is so dense like PNG, do birds call likely in the same spectrogram or, on the same frequency, or are they kind of separating different frequencies? Why is this important? Because it helps with detection, it helps with understanding how species share the soundscape. This is just a bit of how different each species occupy the soundscape throughout sites. I'm not going to go explain this, but this is probably more interesting, this image here. As you can see, on the y-axis is the differences between one species and another species frequency-wise, meaning that if that value in the y-axis gets a bit higher, then the species are quite separate. But when they're getting really a bit lower, that means their frequency bands are really close to each other. And given a really dense and highly diverse area like Managalas over there, it would make sense that they are kind of really close to each other when calling. They need to occupy different frequency bands so they can hear each other. Because if they overlap, imagine we have something called a cocktail party effect. Is that when everyone's talking at the same time, you can't really make sense who is who. And this is the same case with soundscapes and wildlife over there. So what's interesting here is different sites have different partitioning. And this is just a kind of a preliminary output of, this is something that I want to look more into, what actually influences these, time of days, and so on. Okay, so that's just a bit of interesting findings that I got, and now we're going into something called acoustic indices. So we had the automatic recognition that was done by WildMon, but also we want to understand beyond just some species. If the species is not in the database, then likely it won't be detected. And we have bats, we have insects, we have frogs, we have mammals, so much going on. But a majority of what's been detected acoustically has been birds. So how do we look at this without going into really, really deep building recognizers? Because that takes a lot of time and effort. So we work with something called acoustic indices. A bit of the concept of acoustic indices is basically, this is work I did in 2017, 2016. I worked with frogs over at Groot Island, and I looked at a soundscape there on the top left, and I tried to annotate them, and I found annotating an 8-second spectrogram will take me around, it's a 1:8 ratio. So around, if I do the math, like around 3 to 4 minutes to get all the annotations properly. And I got detection of 4 species there, but what happens if I look at a larger scale that looks at 24-hour recordings, then that simple 8 seconds, the spectrogram itself will be around 1.2 kilometers long, and you just do the math of multiplying my efforts. So then maybe you can compress the spectrogram so you can see everything at the same time and go annotate it. It doesn't work that way because this is what you come up with if you compress the spectrogram. It's very clear that you can see the daytime and the nighttime, but that's all you can see, nothing else. You can't see the calls, you can't see anything. Now, thinking of the concept, basically sound is energy. And with energy, it starts with waveforms, and then you can translate it into a spectrogram down there to see what's inside that waveform. But because it's energy, it has so much properties in it that actually we can quantify the different forms of properties to maybe make sense of how we can understand these. So imagine if you're doing landscape analysis, remote sensing. In a GIS manner, you'd have the base map and the many, many layers. You've got the forest layer, the urban layer, and all layers that you can put in, and then you create a map. And that's an Australian map over there. There's a vegetation layer, so you have different types of forests. You don't really know what species is in there into the detail in a larger scale, but you do know that that's the same type of forest. And you do want to change and look at quantify and monitor changes based on how those layers change. So we're doing the same with soundscape. Can we quantify layer? Maybe it's a layer of bird calls, it's a layer of insect calls, it's a layer of bat calls. Can we quantify them and put them in the same logic as remote sensing does, as GIS does? And that's what acoustic indices are. Basically, I'm not going to go into detail, but basically calculating the different types of energies, different types of, sound properties, how complex a call is, that's ACI, how the energy distribution is, that's ENT or H there, entropy index, and then maybe everything else that's considered background noise. And then it creates something called a false color spectrogram. So then something like this that we did see earlier that was compressed becomes something like this. Now you can start to see patterns. Now you can start to see that there's kind of like a harmonics going at the top. And then at the bottom, you can see that there's red stuff that might be a call on nearing the end of the nighttime. On the left side, you can see there's like a double of, let's see if my mouse can do this. Oh, is it good? No, it's not showing. But you can see like a lip double thing, that's also a call. And this has been developed. This has been developed since 2014. And over there on the right side, you can see that that's detecting the calls. How do you detect it? Apparently, even though you look at it on a large scale, the shapes retain. Like the olive-backed oriole down there, which is the lips that I said, they retain the shapes in the larger scale false color spectrogram. The crows, you can find them, the honey eaters. If you look at them on an actual spectrogram, they retain the shape and it's quite distinguishable. For the frogs itself, on the bottom and the top left over there, there's some similarity in patterns. That's an ornate burrowing frog, and then that's I think sedge frogs. I know the Latin name, Littoria phallax. So they're retaining the shape. Again, the color ones are the false colors. You can see on the bottom it's hours. So there's something, it can go like 2 hours that you can see when it starts to call, when it ends to call, but then you can also see on a smaller scale. There's also work on Lewin's rail that also detected similar things. So what do we do when we apply it to a complex ecosystem like Managalas? Having this site, again, grid 38, looking at this area, and this is one site. This is what that one site looks like. It might be confusing at first, but then if you look on the y-axis, it's the spectrogram. So that goes all the way to 22,000 hertz. And then on the x-axis is time. Because I'm used to seeing this, I can already see that that's 3 days. So the horizontal lines, those are night calls, and frogs and everything, they kind of call quite repeatedly, so therefore the energy is quite constant, and the choruses make straight lines, straight horizontal lines. The vertical lines are daytime, which is more dominant by birds, and birds have a burst in sound. They call, they stop, they call, they stop, and that's the type of energy that's being captured up there. If you go up in the higher spectrogram, which higher frequency, which I'm still yet to explore, we don't know yet. It might be insects, it might be bats, it might be birds calling at higher calls. It's still something that I want to explore. But that, another thing that we can see is if we go back, so that's a site in Australia in Samford near Brisbane, which is quite easily to look and kind of identify what's what. Managalas is a complex biodiverse landscape. Every single frequency band is occupied by something. That is how amazing tropical landscapes are. Now, plotting that into several sites is what it looks like. So that's around 1.5 days. If people can see, you can see the nighttime there in the middle, and then the daytime. And every single site looks at a glance similar, but actually there are small differences. Like site 4C, which is in the corner there, is really quiet compared to the site A on the other right corner there. And these small details are what I'm really interested in. How can we capture them, quantify them, and monitor them? How can we do that with something this complex? And with everything that's complex, you break it down. You got one big thing, and then you break it down into smaller pieces. For a soundscape, what does that look like? It looks like you got a complex soundscape, and you break it down by time first. Can we identify distinct time periods? If they are as undisturbed as we hoped, then yes, wildlife species have diel patterns, so likely they will repeat themselves, and likely we can quantify those. And then also, like I said about the partitioning space of the niche partitioning of the spectral space, we can also identify different patterns in different frequency bands. And I've done partly with one site. I wanted to put sound here as well, but the file was too big that it crashed every time, so sorry. If you want to hear it, for those in the room, feel free to come over. I'll be happy to let you listen to it. But we found that they're very distinguished, that every single site, you can actually quantify what the periods are like. This particular site has 5 periods, but some sites have 6 periods. And for this one, night period is very distinct, and it occurs during those times. And after 4 o'clock, it starts to transition to a dawn chorus. And dawn chorus and dusk chorus are actually very similar in pattern, so they're actually clustered together. And then that transfers to a short morning period for around 2 hours. Then it goes directly into an afternoon period. So if anyone's been in the forest and they kind of been in the dawn chorus where it's very busy, you kind of feel that in the morning it slows down, in the afternoon it's not as busy. And apparently it does, there are different times in the soundscape that this happens. And then in late afternoon, things starts to pick up again before it enters the night period. So that's the temporal space. The spectral space gets a bit more trickier on how to do this. So I took one time, which is the dawn and dusk chorus, and figure out, can I cluster these patterns? Because these indices are actually, they're numbers being plotted into an image. So can I take these numbers and cluster them and make sense of them? If landscape images through GIS and remote sensing, you can identify the patterns as in, this is a forest, this is an urban area, this is agriculture and stuff. Can I do the same here? The answer is very much potentially yes, because on the left side over there, you can see that that isn't from indices, but those are clusters. So every single one can actually gather to a distinct cluster. I think this site has 31 clusters. And they, when you plot it, they're kind of really similar to the false color spectrograms, which were layering the indices number. If you cluster them, then you start to actually see these patterns of, for example, on there around 0 to 5,000, you get those blue streaks. That's a cluster of its own. And that's maybe birds. You got the bottom on the kind of the lower frequency part. And as you go up, you've got the higher frequencies also clustering. Hopefully you can see this. But this is what each cluster looks like separately. And on the top is kind of the lower frequency, and it goes higher when it goes down to the bottom, which I know I should have swapped it, but here we are. So yeah, you can see from trait 1 to trait 2, you go all the way from the lower frequency, 2 goes higher, 3, 4, 5, 6, and it just keeps on going up and higher and detecting different patterns. I'm at the point now is trying to figure out what these patterns are. Because the idea is if we know what they are, we know how to quantify it, and bam, we know how to monitor it. Okay, almost finishing up my talk. I just want to talk a bit about some master students that we have on the project and their amazing projects. So first we have Kennedy, who's looking at using the false color spectrogram to monitor frogs. Because a majority of what we found were bird calls, and it was really difficult to find frog calls because PNG is one of the underexplored sites for frogs, and many of the frogs down there are unknown. So his very big task is to figure out how to detect them, identify them, and use them to monitor changes in the environment. Another amazing master student we have is Danny, and Danny's looking at the camera trap side of things. And understanding whether the mammal diversity changes through elevation and distance to gardens or to plantations and such. But another thing that he's looking into is because in PNG, it is community-based forest, but communities still use the forest for many things, including hunting. So he's really keen to know how much hunting influences the mammal diversity there. And that is all. Thank you very much.
Thank you very much, Karlina. Very super interesting. Do we have questions from the audience first, and then we'll see if we'll go to Zoom.
Oh, I just want to also add that if anyone wants to see cool stuff beyond what I'm showing, some of it is in my laptop. So yeah, please do come over, and I'd love to show. No questions? No questions?
Look at spatial distributions. Are you going to look at data on spatial distributions of species?
Throughout if I'm going to look at the data for spatial distribution in the landscape.
That's right. Yes, since you've got the grids and you've got grids moved up.
Oh yeah, definitely. Right now we only have 4 grids, which I feel you can do some things with, but it's not as laid out as 18. And with the 18 grids, it's actually designed that it covers different levels of elevation. It also different geological features, and also distance to, to open areas and such. So there's a lot that can be done, but still waiting on that data set. Oh, within the grids, so kind of inside and then outside. Oh. Yeah, definitely. I did play around a bit with understanding the diversity based on elevation, but not particularly distribution, but very interesting point. I will highly consider that. Thank you.
I have a question, Karlina. So this is really fantastic to see it set up and collecting all this data. And is the idea then that you'll continue that into the future, monitoring in some at that intensity or a subset of that, or what will it do going forward?
Yes, so again, the project, we were actually collaborating with Landscape Alliance and the others. The project itself ends until I've got 2 or 3 years to do this, and then what they're gonna do with the data set will be left up to them. Maybe they'd wanna ask us to look at it more deeply, but we don't know yet. But I personally, because in the beginning I say I work with people in East Kalimantan, and there was the Acoustic Congress happening actually last year, and I've talked with a lot of people, and I say I'd love to use what I found in your sites and see how well that applies. Yeah, so definitely.
Great. Yeah, thanks. Any other questions? Anyone on Zoom? No? Okay, well, please join me in thanking Karlina once again. Thank you very much.
Yeah.